don't even know where to. Okay. So, thank you for joining me today. My name is Ruby Perez, and I will be answering the question of whether dopamine signaling changes in a Huntington's brain. So some background on Huntington's disease is it is a neurodegenerative disease that is fatal. It follows an autosomal dominant inheritance pattern such that when two people have children and one parent has the condition, in this case the father has the condition, there is a 50% chance that every time these two have a child, the child will inherit the gene. And the Huntington gene is the gene that becomes problematic. It has nucleotide repeats, CAG repeats, and when it becomes pathologic is about 35 repeats. When there are these repeats, the gene starts to code the protein incorrectly, and the protein will fold, which forms inclusions in the brain, and it becomes toxic to the cell. And that is what ultimately causes cell death. There are three main symptoms that follow from this. There are motor symptoms, and these can be involuntary motor movements, and the most common is chorea, or Huntington's chorea. There are also cognitive symptoms that occur, such as dementia and problems with working memory. And there are also psychiatric symptoms that occur, which is what I will be talking about today. And these include impulsivity, risky decision making, and the perseveration of these maladaptive behaviors. So, where we're looking at in the brain is along this mesolimbic cortical pathway. And the players that we will be looking at are the nucleus accumbens and the prefrontal cortex, and they both receive dopamine from the ventral tegmental area in the brainstem. To look at this, we decided to use a mouse model, specifically the N17182Q mouse model, and we can use mice because they are genetically similar to make inferences about human Huntington's, and these mice are special in that they have 3 copies of the Huntington gene. They have 2 of their own Huntington genes, but then they have an insertion of a human Huntington gene that is elongated to 82 repeats. And this mouse model was used because they show similar symptoms to adult onset Huntington's disease. So from this, the hypothesis becomes that those with Huntington's disease will have less tyrosine hydroxylase in the mesolimbic cortical pathway as the disease progresses when compared to healthy controls. And now, I just said tyrosine hydroxylase, and you might be like, oh, what are you talking about? So dopamine and tyrosine hydroxylase will mean the same thing in this presentation. However, just to make it as clear as possible, tyrosine hydroxylase is an enzyme that is responsible for regulating the synthesis of dopamine. So the amount of tyrosine hydroxylase is an indirect way of measuring dopamine. But from here on out, I will say dopamine. I do really mean tyrosine hydroxylase. So the methods can be seen on this timeline here. We sacrificed the animals at different time periods, at 5 weeks after birth or 15 weeks after birth. And there's a table at the bottom that shows the numbers within each group. Those groups had wild type and Huntington's mice within them. Then, after the animals were sacrificed and the brains were harvested, we treated the tissue with immunofluorescent techniques. And then we took photomicrographs from the tissue treatment, and we analyzed the photos for dopamine intensity on ImageJ. So, back to our players in this game. So the nucleus accumbens was mentioned, but there are 2 components to the nucleus accumbens. There's the core and the shell. And what is highlighted here is the core. And the core is involved in the recruitment of motor regions to physically go toward a stimulus. And the striatum is also mentioned as a side note because it is also a motor region that helps recruit other motor regions for voluntary movement. And so, we were looking at dopamine signaling and if it decreased, and these graphs show that no, it did not decrease. So the red line over here on the left shows that there was no difference between the 5-week time period and the 15-week time period with core staining. And we were measuring mean gray value, which is basically pixel intensity. And again, there was no difference for the wild type as well between the 5-week and the 15-week. No significance there. And within the striatum, the same thing. There was no difference between the mutants and the wild types, and no difference between the 5 weeks and 15 weeks of the mutants and the 5 weeks and 15 weeks of the wild types. Moving on to the anterior cingulate cortex. This cortex is involved in decision making, emotional regulation, and error detection. And again, it is highlighted in yellow on this coronal slice of a mouse brain. And we actually did see dopamine signaling decrease. There was a main effect for time, such that the 5-week mice groups had more tyrosine hydroxylase, more dopamine signaling than the 15-week time group, suggesting that over time we lose dopamine in the anterior cingulate cortex. Moving on, again, the nucleus accumbens had the core. Now we're looking at the shell. And the shell is this brake system and suppressor of behaviors. It's kind of the second analysis of I need to reevaluate and step aside, step back. And so for dopamine signaling, we also saw a decrease. And we saw a decrease for genotypes. So the two genotypes were different, such that the mutants had less tyrosine hydroxylase, less dopamine signaling at 5 weeks and 15 weeks when compared to the wild types at 5 weeks and 15 weeks, suggesting that there's something about being a mutant Huntington mouse that makes your brain different than a wild type mouse. And again, we were measuring mean gray value, looking at the intensity pixel brightness. So we, just to wrap up again, there was nothing of significance in the core and the striatum. And for the anterior cingulate cortex, time was the only significance that we saw, which might just be due to normal aging. With the shell, however, the mutant mice. Oh. Okay, sorry.
Hi.
No, you're good.
Okay, were you talking?
I was.
Are you practicing?
Yeah, I'm recording myself, but no, it's fine. It's fine. Okay. So, the mutant Huntington's mice had less dopamine than the wild types in the nucleus accumbens shell. And so there are 3 possible explanations as to why we saw this in the shell. So with impulsivity, it has been found that a decrease in shell activity is correlated with an increase in inappropriate responding behaviors. This means that people are impulsive to act. They're too quick to act on certain things, certain behaviors, and take certain actions. With the riskiness, the shell plays a role in learning to inhibit behaviors associated with punishment. And this can kind of be seen with gambling in humans. So, it might be that someone with Huntington's disease heads off to Vegas and they sit down at a slot machine and they're not learning that the behaviors that are associated with losing money are maladaptive to them. This moves into the perseveration of these risky decisions and impulsive decisions. A decrease in dopamine has been related to a continuous Okay, with perseveration, a decrease in dopamine has been Perseveration continues after reinforcers and punishment has been removed with a decrease of dopamine. Did I say that? What the heck? Perseveration can be related to a decrease in dopamine such that these behaviors are continuous after reinforcers and punishments have been removed. So they continue to make these behaviors, and that is correlated with a decrease in dopamine in the shell. Some limitations of this work include time. So time being that this research could be more complete with running behavioral tasks and confirming that these mice exhibit similar behavioral problems with humans. There wasn't a behavioral test run for impulsivity, perseveration, and riskiness. So to complete and make the results more robust, the behavioral tasks would be needed to better make the claim. Additionally, the time that we looked at of these animals. So in the shell, there was a main effect for genotype, but we did not see where the two genotypes converged from each other, or diverged, sorry. We didn't see the starting point when having Huntington's disease made you different. So looking possibly prior to 5 weeks might illuminate some of these differences early on as to when these brain changes happen. And then additionally, with the anterior cingulate cortex, we saw that time mattered, but we don't know, again, when time mattered. So looking, again, before the 5 weeks and after the 15 weeks to see clearly where these two diverge from each other. A next step for the lab would be to look at these receptors, and it might be that we're looking in the wrong place by just looking at the presynaptic terminal releasing dopamine. So with this, we have this presynaptic neuron here that could be releasing dopamine, and the dopamine release might be normal for what is expected, and where the problem might be occurring is with these receptors. So looking at receptors on the postsynaptic neuron might allow us to make more claims about what's actually happening in the brain. So to conclude this all, if you remember one thing from this presentation, is to think of the circuit as a whole. There are many other brain regions involved in this mesolimbic cortical pathway that we did not look at. We just looked at 2. And this can be analogous to a simple circuit. So the simple circuit here, the goal is to illuminate the light bulb. And if your light bulb isn't turning on, you might check the battery and make sure that your energy source is sufficient enough to power the pathway. If it's not, you can replace the battery and then you have the problem solved. But if your battery is fine, you might look at the switch. Maybe you have a faulty switch. Well, if the switch is working, then, okay, what's the next thing that we look at? Maybe your light bulb is broken. Maybe the light bulb that you have is broken. So, again, if all 3 of the things seem to be working fine together, there might be even something else going on. You can see there are different wires. You can see the connections to each battery, switch, and light bulb. And making sure that everything along the way is working together will allow us to make a more clear, will allow us to make a stronger argument that there is a change happening in this pathway. Furthermore, let's say one of your wires is a little broken, but just enough to keep the circuit going. But then let's say the other wire is a little broken, the connection's not as strong. And maybe that in those 2 places where the wires are connected, that's enough for the circuit to be problematic and dysfunctional. So with that, I would like to thank you. Specifically, I would like to thank Zach Daniels and Dr. Mark Pitzer, my lab mates and advisors throughout this project. Couldn't have done it without you. I would also like to thank the McBride Lab at the Oregon National Primate Research Center for the donation of the tissue. And of course the mice themselves, because without the mice, this project would have never happened. And finally, Dr. Zach Simmons for further advice in compiling my notes together. I don't know what to say. Okay.